Greetings, ladies and gentlemen, and welcome once again to today's class. And we want to look at protein synthesis as fast as we can. Protein synthesis, protein synthesis, as fast as we can. I hope you can see the board. Yes, clearly. So, in protein synthesis, as a matter of fact, it's important you understand that protein is so important to the body. As a matter of fact, without protein, the cells cannot survive. The cells are built on this macromolecule that is called protein. I've been trying to, relate this protein synthesis with the drugs we take each time I teach about protein synthesis or I talk about protein synthesis. Most of us have, come down with bacterial or fungal infections. And each time you had this infection, you normally go to the chemist to get drugs, antibiotics drugs. Now, when you take some of those drugs, what those drugs do is that they stop the process of protein synthesis in that bacteria or fungi. And once you stop the process of protein synthesis in a bacteria, or not just in a bacteria, in any organism, then what you are trying to do is to kill that organism or to kill the cells, because protein is an essential part of the cell, and the cell cannot survive without synthesizing protein. And that is why we want to look at what happens during protein synthesis. It is important to understand that there are two different stages when it comes to protein synthesis. The first stage is what we call the trans, transcription, and the second stage of protein synthesis is what is called translation. Transcription and translation. I hope you can see it. Yes, transcription and translation. So we want to look at what happens during the process of transcription and what happens during translation. And let me use the structure of a cell to quickly look at this. Some of the structures that are important in protein synthesis, I mentioned them in the previous class. I mentioned DNA, I mentioned RNA, messenger RNA, and transfer RNA. I also mentioned the ribosome. So, this DNA is found within the nucleus of the cell. For protein to be synthesized, the first thing that happens is transcription. And transcription means that the DNA replicates itself, or a kind of yes, that word, I think that word is the correct word, replicates itself or produces a transcript of itself. And that transcript of itself it produces is the RNA. And this RNA will contain the instruction for protein synthesis. The RNA is working based on the instruction that has been given to it by the DNA. And in the DNA, it's possible that inside the DNA has looked and discovered that the cell needs maybe 20, maybe 10 amino acids. So the information for the production of the synthesis of these 10 amino acids is already figured out within the DNA. And once the DNA goes ahead to transcribe itself in form of messenger RNA or transfer RNA, that messenger RNA or transfer RNA will say contain the same information for the synthesis of those 10 amino acids. That is how it works. So the first thing that happens in the process of transcription is that the DNA transcribes itself into transfer RNA and messenger RNA. Transfer RNA and messenger RNA. Once it transcribes itself into, okay, let me say it before I forget. Please understand clearly that transcription, the first stage of protein synthesis, occurs within the nucleus, is nuclear, whereas a translation occurs within the cytoplasm. So I'm still trying to explain the first process, which is transcription. So DNA transcribes itself into two main types of RNA, the transfer RNA and the messenger RNA. I will quickly tell you the function of these two different types of RNA. So what happens is that the messenger RNA comes. I've already given you the structure of the messenger RNA, having a cap and a tail, and of course AUG, and UAA. So that was what I drew as a structure of as a structure of as a structure of the as a structure of the messenger RNA. So once this transcription occurs, messenger RNA is produced, transfer RNA is produced. So the messenger RNA comes first of all to attach itself with the ribosome. Now understand that I mentioned in the previous class that ribosome is of two different subunits, two different subunits. We have the 30S subunit and we have the 50S subunit. So the first thing that happens is that the messenger RNA attaches itself to the small subunit, that is the 30S, the 30S subunit of the ribosome within the cytoplasm. And once it does that, then the the transfer RNA, of course, goes to a place in the amino acid in the cytoplasm. Sorry, there's a place in the cytoplasm which is called the amino acid pool. Amino acid pool. Within this amino acid pool, you have lots of amino acids. And before, sorry, let me now say what it does, it goes to do. So the transfer RNA goes to pick amino acid. And this amino acid is going to pick will be, like I said, based on the instruction that it gets from the DNA during the process of transcription. And like I mentioned in the previous class, the first amino acid to be produced during every cycle of a protein synthesis is the methionine. The reason is because the cap of methionine contains AUG, and AUG is the codon that codes for methionine. Methionine is the first amino acid to be produced in every process of protein synthesis. So the transfer RNA, when it gets to this amino acid pool within the cytoplasm, it picks methionine because methionine is the first amino acid, and transfer RNA carry methionine, transfer RNA carry methionine is normally written this way, and it says to be charged. You can see the way I wrote it. Transfer RNA met. This abbreviation shows that this transfer RNA is speaking. And it is also important I mention at this point that every amino acid has its own enzyme. The enzyme that has to pick the amino acid and position it on the open end of the transfer RNA. So that enzyme is called amino acid transfer. So let me write it here. The enzyme that helps in positioning of amino acid on the open end of transfer RNA. Amino acid transfer RNA synthesis. Amino acid transfer RNA synthesis. This is the enzyme that helps in the positioning, because I've already drawn the structure of transfer RNA in our previous class. So you can see, once this transfer RNA, this is the structure of transfer RNA. Once this transfer RNA gets to this amino acid pool, then this enzyme called amino acid, I hope that name is correct. Yes, amino acid transfer RNA synthesis. This enzyme, what it does is that this enzyme will now link. Let's assume that this is the amino acid methionine, containing met. So this enzyme will help to link or to position this amino acid on this open end hmm, on this three primer end of the transfer RNA. And once this thing happens, it forms what we call initiation complex. So both the I didn't even mention, there's so many complexes that will be formed here. So the messenger RNA and the small subunit ribosome forms a complex. First of all, forms a complex that is called the messenger RNA 30S complex. 30S complex. 30S complex. So by the time this transfer RNA picks this amino acid, which I said is methionine, and of course, with the help of some initiation factors, it comes to deposit it on the messenger RNA. It comes to deposit it on the messenger RNA. And once it deposits it on the messenger RNA, then the large subunit of ribosome is now incorporated. Let me repeat what I said before. So I said, once this methionine is picked, then the transfer RNA travels from this location, amino acid pool, and where is it going to? It's coming to this messenger RNA in such a way that there will be a complementary base pairing between the codon of the messenger RNA and the anticodon of the transfer RNA. And of course it results in the formation of a bond between the two. The codon of the messenger RNA, remember I said codon is used for messenger RNA, then the sequence of three nucleotides that are found, the sequence of three nucleotides that are found in transfer RNA are now called anticodon. That of messenger RNA is called codon. I hope you have not forgotten this. So the transfer RNA comes to deposit the amino acid on the messenger RNA. So this forms a large complex now. This complex is made up of the messenger RNA, the 30S subunit, the transfer RNA carrying the methionine, and the 50S subunit. So it forms messenger RNA 30S 50S complex. That is what they call initiation complex. Initiation complex. Okay. By the time this 30S and 40 and 50S, these are the two subunits of ribosome. By the time they come together, they now form what we call a functional 70S ribosome. And that means that they are meshed together at this moment. Remember that when a cell is not synthesizing protein, the subunits of the ribosome are dissociated. But during the process of protein synthesis, that is when they kind of come together to form 70S subunits. And these 70S subunits, I'm done describing the initiation. Of course, I forgot to say this. Let me say it here now. There are three different stages during the process of protein. I mentioned two different steps in protein synthesis, that is transcription and translation. And that transcription has three stages, and translation also has three stages. But I've jumped to the three stages of transcription. There is a kind of the enzyme RNA polymerase unwinding the double helix structure of the DNA to synthesize and this happens at the promoter site to synthesize the transfer RNA or the messenger RNA. And so I wouldn't want to go into that. We have steps or stages in transcription, but my emphasis now is to look at the stages in translation, because that is where the actual protein synthesis occurs. So under translation, which is the second step in the process of protein synthesis, under translation we also have initiation. Initiation. After initiation, we have the second one, which is elongation. elongation Elongation. And the third one is called termination. The third stage is called termination. How many minutes now? This is about 14 minutes. Okay, we still have time. So the first stage is translation. The first stage is initiation. The second stage is elongation, and the third stage is termination. So let's go. All the things I've explained so far happens. All the things I've explained so far happen within this initiation stage. the amino acid transfer RNA, helping the transfer RNA to pick its correct amino acids. And that is the function. The function of the enzyme is to enable the transfer RNA to pick the correct amino acid within the amino acid pool. Remember that transfer RNA and messenger RNA were transcribed at the same time from DNA. So both of them contain the same information for the synthesis of certain protein depending on the information that is contained in the DNA. But the transfer RNA may make mistake in picking the wrong amino acid. And that is why every single amino acid has its own enzyme. The enzyme that helps it to pick the correct amino acid and fix it on the surface of the transfer RNA. That's the function of that enzyme, amino acid transfer RNA synthesis. So we are still going. So we are now at the second stage, which is elongation. Within this elongation, that is when the real protein synthesis occurs. Within the elongation, we are told that, let me clean this. Remember I said that to form that initiation complex, we have initiation factors, we have a messenger RNA, we have the subunits of ribosome 30S and the 50S, coming together to form a functional 70S subunit, which is now ready for the incorporation of amino acids. So we want to now get into the real deal, which is the incorporation of amino acid to form polypeptide, which translates into protein. It is important you know that 70S subunit has three different cavities. The first cavity, the second cavity, and the third cavity. This one is called the E cavity. This one is called the P cavity. This one is called the A cavity. E means exit cavity. This is where transfer RNA that has deposited amino acid will exit from. That is through this side. Then this one means peptidyl. It is important you note that it is only the transfer RNA carrying methionine that goes through this peptidyl. Apart from the transfer RNA carrying methionine, which is transfer RNA, met. Apart from this, every other transfer RNA that picks every other amino acid will have to pass through the A, and that A is amino acid site. So these are called sites. We have exit sites. So this is exit site. Peptidyl site. Then we have amino acid site. So these are the different sites. And they have explained that both the transfer RNA met, which is charged. Remember, any transfer RNA carrying amino acid is said to be charged. So the one carrying methionine is called charged transfer RNA met. And it is only this one that has a right to go directly through the P site. Apart from it, every other amino acid will have to pass through the A site. And once they are done depositing the amino acid that they are carrying, there will be a kind of peptide formation. Then they will not exit through the exit site. We'll still come to that. So let me go ahead to explain. So the transfer RNA that carries the methionine comes here. The transfer RNA, it is carrying the methionine, the amino acid called methionine. Remember, it is carrying the amino acid called methionine. It's carrying the amino acid because of space. I will not give the full drawing of amino acid. So this is the E site, P site, A site. So this one is methionine. Like I said, let me repeat it again. I said that the transfer RNA carrying methionine will have to come directly through the P site. But apart from this one, every other transfer RNA carrying any other amino acid will have to pass through the A site, which is amino acid site. So we have now deposited this. Don't forget that messenger RNA is also found beneath this with the ribosome. Sorry, the messenger RNA is also found beneath this because there will be a point of attachment, like complementary base pairing I told you about, between the transfer RNA and between the transfer RNA and the messenger RNA. That is the codon of messenger RNA and the anticodon of transfer RNA. So this this transfer RNA carrying this methionine, once it deposits this amino acid, then it goes, enters into this E site, enters into this E site, and once it enters into this E site, that means that here is not open. Here is not open. Then the second transfer RNA carrying another amino acid comes here, carrying its own amino acid, carrying its own amino acid. I don't know if you can see what I'm saying now. So this one, once it gets to the exit site, then of course from this exit site, it leaves the what's it called again? It leaves the site, and once it leaves the site, it goes back to the amino acid pool to pick another amino acid. Meanwhile, a peptide bond will now be formed between these two different amino acids. And once a peptide bond is formed, this transfer RNA 2, which is now in A site, moves to P site. And from P site, it moves to E site, which is the exit site, where it goes again into the amino acid pool. Remember the amino acid pool, where it goes to the amino acid pool to pick another amino acid. And that is how it continues to pick amino acids until that full number of amino acids that the DNA contains the instruction for the formation of will be formed completely. It will keep going to the amino acid pool to pick amino acids. But then, it is also important you know that there is usually a delay during peptide bond formation. That this delay is not just to ensure that some proteins called elongation factors ensure that the transfer RNA carrying the second amino acid is correctly positioned at the A site. That is one of the reasons why there is a delay, or one of the benefits of the delay. It ensures within that period, the elongation factor will ensure that this transfer RNA carrying the second amino acid is correctly positioned on the A site, because it must pass through the A site. But then the most important reason why there's a delay is to make sure, or to checkmate, that the transfer RNA is carrying the correct sequence of amino acid. Remember that the sequence of the amino acid must begin with AUG. And let's assume that the second one is phenylalanine, which is UUC or UUU or UUC. Let's assume. So there is what we call editing of translation. Translation is edited through what is now called proofreading. So within this period of delay, there is a kind of a system that checks if the transfer RNA is carrying the correct sequence of amino acids that the messenger RNA contains the information for. So the information that the messenger RNA contains starts with the formation of methionine AUG. So the transfer RNA, it will be checked if the transfer RNA is carrying that AUG amino acid for the first amino acid to be synthesized. And like I said, let's assume that the second one is phenylalanine. Phenylalanine is UUU or UUC. So within that point of delay, before a peptide bond is formed between this methionine and phenylalanine, then that delay occurs to check if this thing is correct, and to check the one that the messenger RNA is carrying to make sure if it's correct. The essence of this delay and proofreading and editing and the rest of them is to make sure that there's no mistake, because If a single mistake is made during this process of protein synthesis, it can signal doom for the cell. And that is why all you notice this delay. So this thing continues to happen until a long polypeptide is formed. Until a long polypeptide is formed. And not just formation of a long polypeptide, until the number of amino acids that has been encoded in the DNA and that the transfer RNA is carrying the instruction for, and not just transfer RNA, the messenger RNA is also carrying the instruction for, are completed. Once these amino acids are formed, then something will now happen. What I just finished explaining is the second stage, which is elongation. The third stage is termination. And it is important you notice that, or you know that, remember the first, there are proteins that help in this protein synthesis. During the initiation period or stage, I mentioned initiation factors. There are proteins that also help in this process. Then the second stage, I also mentioned some proteins that are called elongation factor. Then there are also other proteins which I've mentioned under this termination stage, which is called the release factors. There are also proteins that help. So when it's time for the termination of the process of protein synthesis, I've mentioned that a cell cannot indefinitely produce protein or anything at all, because the product of a cell can kill a cell. And that is why there is turn on and turn off mechanism within the cell that switches off the process of protein synthesis. And once it is time for this process of protein synthesis to be switched off, then what happens is that a nonsense codon will be encountered. And I mentioned those nonsense codons. Those nonsense codons, you are, or ag, and or ga. These are nonsense codons. Once these nonsense codons are encountered, there is no transfer RNA that contains the correct anticodon that will bind with the transfer RNA. That will bind with the nonsense codons. And because this thing does not occur, or because transfer RNA does not contain the anticodons that correctly binds with the nonsense codon, then the release factors, these are proteins, will now mimic the transfer RNA to bind with these nonsense codons. And once they bind with the nonsense codons, it results in cleavage. That cleavage signals the termination of the whole process of protein synthesis. So in summary, encountering nonsense codons means that process of protein synthesis is has finished and should end at that moment. Not even about to end, but should end, because the cell is done producing the number of protein it requires or it needs at that particular time. Then this signals the termination of the process of protein synthesis until when next the DNA feels it's also necessary for protein to be synthesized within the cell, then the process is switched on again and it continues. So I believe, or I think that this will go a long way in explaining or demystifying this mystery behind protein synthesis. I noticed that students find this protein synthesis very difficult, but with what I've explained so far, I'm uh, I'm hopeful. I hope that it has explained that. What's the time? Okay, this is 29 minutes. Okay, so there are some biomedical applications of protein synthesis. This is mainly for medical students. Medical students will have to, pay attention to some of this, and I will read out some of these, uh, some of these, some of the applications of biomedical applications of protein synthesis. It says protein synthesis can also be stopped by chemical means through the administration of antibiotics. I know I've mentioned this, that by the time you take antibiotics drugs, uh, then you are trying to kind of stop the process of protein synthesis. And once you stop a cell from producing protein, that means you are about to kill the cell. It says about 160 antibiotics are known and they have been extremely important in fighting diseases. This is by virtue of the fact that they impede the process of protein synthesis in bacteria, often poisoning bacteria selectively. Chloramphenicol, chloramphenicol, that is a drug, and antibiotics. You can see what some of these drugs do, like I explained. Let's just look at them one after the other. This chloramphenicol, it helps to prevent peptide bond formation. So this peptide bond, formation of this peptide bond, this drug prevents this bond to be formed. And once this peptide bond is not, once the peptide bonds are not formed, or once the peptide bond is not formed, then of course you cannot synthesize protein. Protein cannot be effectively synthesized because the synthesis of protein is a product of this peptide bond that is formed to produce a long polypeptide chain which translates into protein. So he said erythromycin, another drug, prevents translocation of transfer RNA from the P site. There's usually a kind of movement of transfer RNA with a kind of there's usually a kind of movement of transfer RNA. Let me just a minute. There's usually a kind of movement of ribosome with respect to the transfer RNA and messenger RNA. So the ribosome is situated here. Once of course you know that I don't know how to explain this now. The codons are found in sequence. So we have first methionine, uh, we'll have second phenylalanine, we'll have third maybe serine, we'll have fourth maybe tryptophan. So these are found in sequence. So the ribosome will also be moving. Once peptide bond, once complementary base pairing occurs between the transfer RNA and messenger RNA, and the peptide bond is formed, then it shifts a little. It shifts a little until this addition of amino acid continues to form this polypeptide. So the movement of ribosome with respect to the messenger RNA and the transfer RNA is what is called translocation. And translocation is important because if there is no movement, then other amino acids will not be incorporated. So you can see that erythromycin prevents this translocation that I just explained now from the P site. Then another antibiotic called puromycin and tetracycline can pose as amino acid transfer RNA and bind to the A site to accept amino acid chain from the transfer RNA at the P site, thereby stopping elongation. Remember I mentioned amino acid transfer RNA synthetase. This is an enzyme. So we have a drug, tetracycline, that can pose as this enzyme. And once it poses as this enzyme, then it prevents the correct positioning of the correct amino acid on the transfer RNA. And this again will affect the process of protein synthesis. As a matter of fact, all these things we are mentioning, if there's any issue in any of the stages, either from the ribosome that should translocate or the incorporation of the two subunits of the ribosome, or probably the attachment of the messenger RNA on the ribosome, or the correct picking of the correct amino acid, if there's any mistake in any of this, then there will be a problem in the whole process of protein synthesis. So you can see the drugs are selective in attacking one or two different components of the entire process of protein synthesis. So streptomycin can bind to 30S subunits to inhibit initiation of protein synthesis. You can see that one. Streptomycin can bind on the 30S subunit. So instead of messenger RNA binding on this, that drug, streptomycin, it binds on the 30S subunit, and it mimics the messenger RNA actually. And that again will stop protein synthesis. Where initiation has begun, streptomycin will prevent uh, messenger RNA from binding firmly on the 30S subunit. So even if initiation has occurred, then it will prevent the messenger RNA. It will prevent transfer RNA from firmly binding to the 30S subunit. The rifampicin, I think that's the correct pronunciation. Rifampicin is also an antibiotic. It says it's used to treat tuberculosis. This antibiotics drug is used to treat tuberculosis. What does it do? It helps to bind bacterial transfer RNA polymerase enzyme. It helps to bind bacterial transfer RNA polymerase enzyme, thereby inhibiting the initiation of transfer RNA synthesis in the bacteria. So you can see what this one does. So it helps, it will now inhibit, the initiation of transfer RNA synthesis in the bacteria, because the polymerase, of course, helps in the synthesis of the transfer RNA. And once it mimics that or inhibits the function of the polymerase, transfer RNA polymerase, then of course it will inhibit the initiation of the transfer RNA polymerase in the bacteria. Then poisonous mushroom contains amanitin, an inhibitor of human RNA polymerase, thereby blocking transcription of DNA to RNA. So this component, this stores in mushroom, will definitely, in human, will definitely prevent the transcription of DNA to RNA. Then actinomycin D, used in cancer therapy, and intercalates between the GC base pairs and DNA, that thereby inhibiting transcription. So these are some of the drugs and some of the specific roles they play in trying to inhibit the process of protein synthesis. We also have energy budget for protein synthesis. Protein synthesis cannot occur. As a matter of fact, protein synthesis is an enterprise, a cellular enterprise that requires high amount of energy. And if energy is not produced, if energy is not within the cell cannot, and mark my word, cannot go on with the process of protein synthesis. They will have regulation at the translation level. This prevents transcribed messenger RNA. Oh, there are so many things here. Those that affect the ability of ribosome to attach. Okay, various control mechanisms are possible at this level and include, we have those that affect ability of ribosome to attach to messenger RNA. We're still talking about the same thing. Those that affect the lifespan of messenger RNA molecules. We also have those that affect the presence of different factors. Those factors are very, very important. So we have the drugs or some chemicals that affect the presence of those factors, the IF factors, that's the initiation factor, the EF factor, that's the elongation factor, and the RF factor, that's the release factor. So some of these can also prevent the functioning of these enzymes. Then we have those that affect the kind and amount of transfer RNA molecule carrying the right anticodon necessary for translation. For example, some viruses produce enzymes called nucleases that destroy transfer RNA molecules of their host, thereby blocking messenger RNA translation in their host. So this you see in virus. Once they produce that enzyme called nuclease, then of course it destroys the transfer RNA molecules of their host and it blocks messenger RNA translation in that host. So that is it. So ladies and gentlemen, I think this is where we'll stop. We'll have another thing to talk about, and that is mutation. But I don't think it's something we can do now. Let me see what's the time. How many minutes? Okay, 39 minutes. Let's quickly see if we can talk about mutation. 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 Can you see the board? Okay. Now, when we talk about mutation, we mean alteration in gene or changes in the gene. That is what we know as mutation. It was coined by De Veres. or Hugo de Veres. Hugo de Veres. Hugo, I think Hugo de Veres. So the scientist that championed this name called mutation is the man they called de Veres. I think there is de Veres, and I think his correct name is Hugo de Veres. I'm not too sure, but either de Veres, I think there is Hugo de Veres. So this is the man that worked extensively on mutation. And like I said, mutation is changes, alterations in gene is what we call mutation. This mutation can occur through insertion, through insertion is same thing as addition. Insertion or addition. Then we'll have deletion, we'll have substitution. So these three types of mutation are called the point mutation. Now let me see. Can you see the board? Okay. Let me see if I can use